welcome to bioanalysis. Previous lecture we are talking about chromatin immunosprecipitations, co-immunosprecipitations and general mechanism of immunosprecipitation. Today our topic is RNA immunosprecipitation. What is RNA immunosprecipitation? RNA immunosprecipitation is a technique to precipitate target RNA protein complex from other RNA protein complex. For use, this technique is to identify which RNA sequence interact with particular RNA binding protein of interest in vivo condition. It can also used to determine relative levels of genes expression, to identify its substrate RNAs which may be co-regulated, or identified RNAs that may have related function. This technique provides insight into the post-transcriptional gene regulation which occur RNA and RNA binding protein. Now I am talking about RNA immunoprecipitation technique. First, cell are fixed with the formaldehyde. It acts as the reversible protein-RNA protein-protein cross-linked agent that serves to fix the protein-RNA interaction occurring in the cell. Then cell are lysed using either mechanical or enzymatic digestion and RNA is released. Then antibody are designed, the protein of interest. Then immunoprecipitant are added, the protein of interest. This design antibody and immunoprecipitant or bead bind with each other and form antibody bead complex. Then design antibody is attached with the target RNA protein complex and precipitate it. This RNA precipitation is mainly two types. One is AGO2 type and other is ABP type. When microRNA attached with the microRNA have some protein like AGO2 which also bind with mRNA and form microRNA rise mRNA complex. After this forming microRNA rise mRNA complex, we are design a antibody which bind with AGO2 and some bead are prepared which also bind with this AGO2 binding antibody. Then this antibody and bead attach with each other and this AGO2 antibody bead complex bind to this AGO2 protein and precipitate this RNA. Other side, messenger RNA have RBPX protein with ribosome related site. This RBPX protein of interest, we are design some antibody which are anti-RBPX antibody and anti-RBPX bead. This anti-RBPX antibody and anti-RBPX bead bind with each other and form anti-RBPX antibody bead complex. This anti-RBPX antibody bead complex bind with RBPX protein of messenger RNA and drag down and precipitate it. In this process, RBPX type of precipitation occur in mRNA.
Thank you. Please subscribe.